Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I'm on behalf of Comstech and myself, I welcome you all to an exciting webinar organized by Comstech. This webinar will be delivered by Professor Dr. Adnan Memik. Dr. Adnan Memik is a professor of nanotechnology associated with King Abdulaziz University, Kingdom of Saudi Arabia. He is also an adjunct professor at University of Connecticut, USA. The title of his topic today, of his webinar, is Translating Micro and Nanotechnologies into Medicine. And to welcome and introduce our guest speaker, I would request His Excellency, Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech. Over to you, sir.
Bismillahirrahmanirrahim. Thank you very much, Hazima Muazzam, for arranging this very important webinar on a topic which is extremely relevant to the modern development. According to McKinsey's report 2019, new material development and nanotechnologies are among the 12 most important disruptive technologies which will have a huge impact on the landscape of the world, and that they would generate tremendous amount of fund through the new applications. And today's webinar is largely focused on the application of micro and nanotechnologies in the field of medicine. I would consider this as one of the most important landmark in the recent history of medicine, as we are moving forward to make currently available medicines more effective, more target-oriented, and more patient-friendly with the new technology. And for that purpose, we have invited a very eminent scholar from King Saud University. And Adnan, I would like to tell you that I've been associated with King Abdulaziz University, with King Abdulaziz University since last 2 decades as the highly cited scientist. And I know that this institution represents excellence, and there are a lot of very high quality research centers which are established. So we have with us Professor Dr. Adnan Memik, and he is very well known in his field. And let me have the honor of reading his CV briefly, which I'm sure that is worth really spending a lot of time. Over the past 10 years, Dr. Memik have been directly involved in the establishment of high-quality nanotechnology research center in King Abdulaziz University with over $25 million of investment in infrastructure. And that was right from the scratch. So it is a world recognized facility now. And that the research in this new center is cross-disciplinary and multidisciplinary, spanning over various categories of material, including biomaterials, polymer chemistry, nanotechnology, resulting in over 100 publications to his credit. 20 provisional and granted US patents are also been obtained by Dr. Adnan Memik. He joined King Abdulaziz University in 2010, instantly being academically promoted to a full professor of nanotechnology. He previously held various positions at premier institutions, including lecturer of medicine at the Harvard Medical School, a teaching professor at the University of Illinois at Urbana-Champaign, and currently as an adjunct professor at the University of Connecticut. He is recognized as a highly cited researcher for 2 consecutive years, giving only 0.1%, 1 in 1,000 of scientists in the world, and as a top 2% scientist by Elsevier and Stanford Analytics rating. He also served as a consultant to venture capital firm, providing technical and scientific advices that specialize in early stage investment in healthcare and life science based companies. Finally, he is also a founding director of the startup medical device. Most importantly, we're very proud that young people like Dr. Adnan are working in these disruptive and emerging technologies, and that really give us a reassurance that the UIC world would also excel in science and technology and will at some point of time reclaim this renaissance of Muslim civilization and Muslim contribution in the field of science. With this, I would like to invite Professor Dr. Adnan for his presentation.
Thank you, Professor Iqbal. Thank you so much for that very, very kind introduction. Very, very generous. I think I'm a little bit humbled by all the stuff that you mentioned. But I'm very much honored to have the opportunity to present. I very much am happy that you gave me this invitation, and I hope that the seminar will be interesting to a lot of the attendees. I've been kind of scrolling through the chat messages and looking at all the different people saying where they're from, what their backgrounds are. So that's very cool, very interesting. So I'll try to make it very very relevant to what what they have been doing and stuff like that, and we can try to see if I can share some of the stuff that we're working on at King Abdulaziz University, right? So let's just start, right? So just where am I from, right? I think Professor Iqbal, he mentioned some of the places that I was. He mentioned King Abdulaziz, in general, but specifically the Center of Nanotechnology, where I do most of my work. It was established in 2007, so we're not that old, so not a lot of people knew about us, but I think as time is going, a lot more interesting work is being published, and we're drawing a little bit more attention to what is going on. It is headed by the Professor Ahmed Al-Shahri. He's our director. MashaAllah, he's a very, very nice guy, very good guy. Very happy to have him as the director. I got to give him a little bit of props because a lot of this work, it's so important to have a, director that shares your vision. And, we have about 15 full-time staff and researchers that work in the center. So we have a lot of different facilities. If you're in the area, maybe you're going from Umrah or something like this, and you would like to stop by, I'm sure that you would be very much impressed. We had many people come visit from MIT, and they're like, "Oh, you have the the XPS, and it's right here," and all this stuff. So I think that's also something very important. But okay. So briefly, I want to give you the outline of my talk. Right, before we get started, I'll give you a general introduction. Not too much. Something more motivational, and then we'll get into what is really nanotechnology, why we're using nanotechnology, and then I'll focus on two really different topics. One topic is about tissue engineering, and the other topic is more about immunotherapy, and specifically, we will focus on bone tissue engineering. So we'll look at the different hierarchies. So we'll look at, gels and cells, and we'll look at something issues with tissues. So you can think about how really nanotechnology can be really used in different hierarchies. And you will see from the introduction how the properties of nanomaterials are very unique. And then as you go through different scales, you can really affect different changes. So you can affect changes on the cellular level, but you can also affect changes on the tissue level. So that's very important to think about as we go about this. And then the oxygen releasing cryogels, we'll talk about that as well as we get along. But I think something which is more important, is the what ifs. I think this is something which is quite overlooked. I think if you think about what is the motivation, why you're doing these things, like, can we take a dysfunctional part of a tissue? Can we remove it? Can we replace it? Can we put a bioengineered machine to take its function? Can we think about, delivering the right medicine at the right time, at the right, location, at the right dosage for the right patient, right? So these are the types of questions that you really want to think about when you're doing your research. And this is very common, right? As a student, especially when you start off your kind of your path on this, the scientific path, the way that you have been usually evaluated has been based on the quality of your answers, right? So you take an exam. You get all the questions correctly, you're a good student. You're an excellent, student to have in the lab, and everybody's like very happy. But when you start doing research, things actually, you know, they start to switch, you know. And this is something that, you know, Bob Langer, who's a famous scientist at MIT, kind of mentioned, and I kind of take this really with, you know, really take it, you know, hold of it. And I really think about this as an important point to mention, which is once you start doing research, it's really the question that you're asking that will really derive the type of research that you can then generate, right? So the question becomes more important than the answer, right? Because you're doing something new. You're trying to find something which has not been discovered before. So if you ask a question very carefully and, you know, very, you know, you study that well, you understand the fundamentals, and this will be some, you know, something that I will mention as I talk about, you know, really knowing your fundamentals is what's important, right? So the question is very important here, and this is a different type of mindset, right? So you have to really, when you start doing research, you kind of have to switch your mindset from this kind of a student mindset into a researcher mindset. You have to kind of change your hat and say, you know, well, what kind of questions can I answer? And these things, you know, when you look at them, you know, they might sound, you know, they sound, you know, science fiction-y, you know, type of thing, you know, pump the right medicine, the right time, at the right location. But when you start looking at the, you know, the literature and things that are being published, this is, you know, the lines start to blur a little bit more and more. The lines become, you know, unclear whether this is something which is really, you know, so far out, or is this something that we can actually do in the lab? So I always like to mention that as a motivation for students, because, you know, the way that they approach their work really can impact, you know, the outcome, you know, whether they're just kind of doing this, going through the motions and mixing things, or really thinking about what they're actually doing, what is the goal, what they're trying to achieve. Okay. So I kind of switched gears, right? That was like kind of the introductory part, but, you know, what is really nanotechnology? You know, and I think in general sense, the nanotechnology, and why do we work with nanotechnology, has been, you know, it's a science or maybe engineering at 100 nanometers or less. So people say like, hey, why did you say 100 nanometers? I mean, you have liposomes and all these different things that are maybe 200-300 nanometers. I mean, it's still nanometers. It could qualify. So if you ask a biologist, they will tell you, you know, it's really has to do with, you know, targeted drug delivery. You know, if a tumor, you know, is getting bigger, you will not have targeted drug delivery if the particle size is more than 100 nanometers. A lot of published work out of this, you know, like Dane Wittrup's lab at MIT and stuff like this. So, you know, valid point, right? If you talk to chemists, they'll tell you, oh, this has to do with the secondary bonding. It has to do with all these different, you know, different way how things are, you know, conjugated to each other. And if you ask a physicist, you know, they will tell you it's 100 nanometers or less because of the speed of light. And that's true. It's so interesting that it's true, you know, and I always leave that question at the beginning. If you really want to find out why is the speed of light the definition of 100 nanometers, you can ask me at the end, but it's very interesting. But really, where did nanotechnology take off from? The nanotechnology took off, you know, relatively in 1959, when Richard Feynman, famous scientist, Nobel Prize winner at Caltech, he established this kind of a Nobel, he established his own prize, you know, like, why not, you know? So it was like two ways that you could win this little prize. There's other prizes now that are named after him, but his original one was that, you know, you had to make a small little motor that could fit, you know, into, you know, like a 1/64th of an inch into, you know, cube, and, you know, work. And it didn't take long for that to really, you know, to that prize to be claimed. It was maybe within a year or so. But, you know, the second way that you could win that prize was if you would print a page of the encyclopedia, let's say, on a tip of a needle. So it had to be like 25,000 times scaled down so that you can get those letters and read it with an electron microscope. And it took long. It took really 30 years. And it wasn't until the 1980s until somebody was actually able to accomplish this. And this is really where, you know, nanotechnology starts to take off. You have a lot of these other Nobel Prizes, like for AFM and stuff like this, that were really based on nanotechnology. And you could start to see that really new properties start to emerge at the nanoscales. So you will see that the size of a particle, the shape of the particle, all of this plays a very important role. And this is, you know, the famous slide that Chad Merkin from Northwestern always likes to present. So I give him credit, but I really like it so much I had to, you know, present it here. But, you know, it really helps you visualize that, if you have silver particles, silver nanoparticles that are spheres versus if they're silver nanoprisms, even if the size is almost very similar, you can see that the color change is quite different. And even you can have gold. If you have gold spheres that are 60 nanometers, they're green. So gold is actually green. So this really kind of helped people understand that, once you start changing the particle size and shape and things of that nature, you really start to fundamentally change the properties of that material. And that's really where you can start to generate novel functionalities. You can start to generate novel materials that are able to serve and to do things that otherwise you thought wouldn't be possible in their as their bulk counterpart. I think that's something which is very important to think about. So how do we, where do we think about this in terms of, tissue engineering and regenerative medicine? Where do we find this, nanotechnology? Where is that niche where you can fit that in? So in general, let's, if we just think about this, in terms of tissue engineering, if you look at the cells in your body, right? Most of the cells, where are they? They're moving around, and they're not moving around, except for your blood. All the cells are kind of stationary, right? So you have something which is holding those cells in place, which is, we can call it the extracellular matrix, right? The cells kind of deposit this little bit of collagen, the softy things, and kind of keep the cells in place so they don't move around. So that's the two things which are obvious, right? You have something which holds the cells in place, and you have the cell itself, right? And you can try to mimic the different parts of, you can try to mimic those kind of characteristics, but there's also regulators, right? So the things that we're all, a lot of people there that, kind of comes to them intuitively is they think about growth factors, right? Cytokines, all these different things, these are kind of a biochemical signaling, right? Often, very well covered in the literature. But when you start to think about it, there's actually other ways that you can affect and change cellular behavior. And this is the famous, paper, I think maybe 2005 or 7, from the Disher lab at University of Pennsylvania, where they took stem cells, and they let those stem cells grow on different types of substrates. They changed only how soft that substrate was. So if that substrate was very soft, they would go into a certain lineage. If that substrate was very, stiff or very hard, then you would have them commit to a different lineage. And there was no other biochemical signaling present. So that was very interesting. It was so there's other ways other than the biochemical stuff that can affect stem cell, behavior. And they were like, hey, wait a second. Is there a way that we can now start exploiting this? Can we use materials not only as the matrix, as the scaffold, but also as something which would be indicative, something that could help the cells, go into the function that you want. And I make this, very, kind of a short digression about, what the difference is between tissue engineering and regenerative medicine. Just the goal of one is more to, regenerate in vitro, and then you kind of have that all, you're engineering that tissue, right? And then you implant it into the body. Whereas regenerative medicine, in general, it's more thought of something that the regeneration happens in vivo, right? You implant the biomaterial. Maybe there's cells, maybe there's not. Maybe there's, other factors or something like that. But really, the regeneration is intended to happen in vivo. So that's something very important. And this is, kind of a, this is really, this is kind of a main slide that I really like to focus on. Other than my what if slide, this is the type of slide that I really like to, think about. Because oftentimes the students, they come to me and they say, hey, professor, we saw this paper here and they did this work with this polymer, and we saw this paper here and they did, this nanomaterial. Maybe we could, combine those two things together and we can get a paper out of it. And I'm like, well, why would you really want to do that? Why would you really, what is the benefit of just combining two different things together? And I see this very, very often, right? Where's really the innovation coming from out of this? What problem are you solving? So this is really where you kind of have to put yourself into a little bit of an engineering mindset and ask yourself, well, am I really solving a problem by combining these two things together? Or am I really just doing some type of a synthesis and characterization type of work? There's nothing wrong with synthesis and characterization. You can, it's important, right? You want to be able to make what you're saying that you're claiming that you want to make. You want to be able to characterize it, which is good. But really the application part, and this is, this is where the why comes from, right? What is the application of this, of these, kind of things, putting them all together, right? And this is, why I always, bother my students, and I tell them that's why it's so important to understand the fundamentals, because that will help you understand where is that innovation coming from, right? You often find this, like many people, they talk about, in different, parts, not just in science, like, oh, innovation, it's so important. We have to innovate, and we have to, do all these different things. And I always say, well, innovation is very simple. It's people. It's people. You can have the, the latest computer, or the latest, I don't know what tool you want to have in your lab, the best equipment. If you don't have a person who knows how to operate that equipment, it doesn't really matter, so that's why really the technology transfer, it really happens by people, putting them into this, into these positions, and understanding why they're doing the things that they're doing, right? So not just the technician, just mix and match the synthesis and characterization, but really understanding why. And you really can't get to that point unless you understand what has happened before. So, and the understanding what has happened before is understanding your fundamentals. Why does this nanomaterial change color? Why would that be important? How could that help me with detecting a pathogen? How, will that make it, better at detecting the pathogen? Maybe I can, detect it at a much lower concentration. What are really the changes that are happening? And this is really, once you start thinking about this, it really helps you. It really helps you kind of advance your research into something which is going to be more impactful, something that is actually, is not only has the application, but is going to have, high impact implications if you're able to do this. And this is kind of a, it's a little bit risky, right? So you have to understand, there's that, that's why I don't say just doing synthesis and characterization is bad, because you need to show that you have those skills, which is very important if you want to go into some type of a industrial, let's say, field. You want to go into industry and work in the industry. It's very important to demonstrate that you have certain skills, that you're able to do this, that you understand why you did these experiments. That's why you often getting your advanced degrees, right? So not just showing that you're able to, pipette and use this instrument, but you understand how to modify those, conditions so that you can generate the outcome that you want. That's really this is what your thesis is about. This is what your hypothesis is about, and understanding those things. But that's really what drives your innovation forward. And once you have that innovation, it's like, well, why would somebody fund my idea? You have to always think about this. Especially as a professor, doing things is so important. But really, it's about finding funding. And so there's the these two books that I usually like to just kind of just screenshot them. Is Sand Hill Road. The Secrets of Sand Hill Road. So if you haven't heard of Sand Hill Road, this is essentially the capital of venture capitalists, right? All these investors, right? It's like the Hollywood. The Rodeo Drive is to Hollywood, or the Magnificent Mile in Chicago, or the Saks Fifth Avenue for shopping. Sand Hill Road is where all these venture capitalists are. And and this is why I say innovation is so important, because the first thing they ask, the first criteria, whether we're going to fund this idea, we're going to put in thousands or millions of dollars into it, is who's on the team, who are the people, what are their skills, do they really know what they're doing, do they understand the stuff? And then they look at the product. Let's look at now the product that they developed. Let's look at the things that they have actually done. Is this something which is innovative? Is this something which is really going to make a difference? And then finally, they look at the market size. It's like, okay, you might have the greatest product, but if the whole, in the whole world, they, only sell 1,000 of these, well, I don't know if we're really going to get our, investment back, because it's like, how much of these can you sell to make it worthwhile? And this is a problem. honestly, this is a problem, especially for, diseases which are very rare. You don't find pharmaceutical companies investing in them. They don't want to. They're like, yeah, it's not really worth it for us. But, that's why there's other mechanisms. That's very important to work on these things because there's other benefits to it. And the other side, of, kind of this innovation and stuff like this is really great innovators. They procrastinate. Don't think, like, oh, this guy, he has it all figured out. It's so easy for him. Writing papers is like, with his eyes closed, that, just coming out, all these great ideas. No, really, you find that you have a lot of bad ideas, and that's okay. It's really having that strength to push forward after failure. That's really what differentiates a good scientist, somebody who, who has a lot of potential versus somebody who's going to, give up on this. Okay, so enough. I took maybe 10 minutes, 15 minutes of your time just talking about these general ideas and all this cool stuff. So now we'll actually get into the, the real stuff that is happening in the lab, right? And really there's 3 different directions for the lab. So really 3 different directions. The first direction is this regeneration of bone. And we'll see why that is important. You can think about cancer resection. You can think about, traumatic injury. You can think about, maybe congenital defects. You can think about, different ways that these that you would need to regenerate bone. The second part of it is like this injectable biomaterial. So very important, thinking about ways to minimally in some type of a minimally invasive method deliver the medicine that you want at the right place, right? You don't want to do surgery. You don't want to do all these different things. Can we do this by making something injectable? So that's something very important. And the third part, and I don't think I'll have time to cover this, is something that we call, a little bit about drug delivery. So it's like thinking about, so I'll just give it 1 second here since I really don't think I'll talk about it later, is developing a, fully biodegradable scaffold, something which has electrical circuitry on it that could, maybe let's say heat the little scaffold, and then the drug that is encapsulated inside can be released, right? So because of that heat, the drug that is in some type of a, gelatinous state will melt, and it will go into the liquid state and it will be released. So that, so you can see really here that, delivering the right drug at the right time at the right dosage is not that far away, because, systems like this, these smart types of systems can really allow you to have a lot of that control, right? So let's look at the bone tissue engineering. What is the motivation of doing this stuff? So if you have a, if you have a small fracture, or something like that, incomplete fracture or simple fracture, yeah, you probably just need a cast. There's nothing there. You don't probably need much more of an intervention than that. We're talking about 175 million. I think the Lancet reported 176 million fractures occur annually, right? So a very small portion of those, maybe less than 1%, maybe less than 0.5%, they start to get more complicated, more compounded, multiple fractures. Now things here are not so easy to solve. Or even sometimes you have a regular fracture and it doesn't heal. So you have to do some type of an intervention. Maybe it's from cancer, maybe it's from traumatic injury, maybe it's from osteoporosis. It could be different reasons for this. But now you're starting to see that there's a way that you need to help that bone regenerate. You need to maybe do some type of an intervention that could help, speed up that process. So the way that we thought about this was to do some type of an implantable, material. So we wanted to build these nanocomposites, these hydrogels that could be used for bone repair. We wanted them to be kind of injectable. We wanted them to be a little bit soft. So now we're talking about cells, right? Looking at what things are happening on the cellular level and how can we modify that to increase or to kind of speed up this healing process. So what we did here is we mixed gelma, which is gelatin, which is combined with methacrylate, which is just a chemical moiety that cross-links that gelatin into a solid structure, right? A more solid structure, viscoelastic structure that can absorb a lot of water. So that's what hydrogels are. They're just a polymer network. You can imagine it like a building, right? And the air inside of it is water, and the walls of the building are the polymers. So this is really what a hydrogel essentially is. But if we throw in these nanosilicates, laponites specifically, they have a unique they have a unique structure. They have very, negatively charged sides, and then you have, positively charged, I think, the rings around it. So this kind of a this distribution, this charge distribution, you start to generate, unique properties. And what you can do is that essentially with time, you can get that stem cell to commit into a osteo, into a bone type of a lineage. So you can take a stem cell and get a bone cell afterwards. So, typical, now you're getting into your, synthesis and characterization type of stuff, right? You're combining your nanosilicates, you're combining your gelatin, you're putting the UV, you're putting some light, something that's going to cross-link this. You have some, photoinitiator that's going to generate this chemical reaction, right? You're going to generate your nice little scaffolds, they're porous. Then you start looking at different types of, different types of characterization methods. Do I really have what I think I have? So you can imagine doing, let's say, something as an FTIR or Raman or XPS, or you can really choose the technique that is available in your lab. But essentially, you have to think about each one of these kind of steps as an idea. Well, the first part is, did I really generate the idea that I'm talking about? Did I really generate, this composite or not? So you can look at the FTIR, okay, here's one peak, here's the extra peak. So when I combine the two things together, I have what I claim that I have. So that's something which is important. So you have synthesized it now. Now let's look at the properties, right? Okay, so I'm proposing this for some type of a bone application. So perhaps mechanical properties will be something important. Perhaps degradation will be something important. And this is where you look at the literature, you look at the different things that are happening to see whether these types of characterizations would be relevant, right? And you see, okay, so I'm getting things which are improving. I'm adding more of the nanoparticle and I'm getting improvement in the properties. Great. But really, I want to do this. You always have to keep that application in the back of your mind, because you want to make sure that you're actually able to use these things. And why is that important? Is it's important because you want to make sure that there's some type of a non-toxic effect by adding these nanoparticles. And you can see that once you start adding more and more of these nanoparticles, you actually can start to make things a bit more toxic. So now you have something maybe where you started off with 20, 30 different particles, or 20, 30 different, composition, and you start to filter them down. Am I improving the properties that I want? Am I getting the functionality that I want? So I don't want it to be toxic. Oh, now if I add too much of this particle, actually I'm starting to kill the cells. So that's not good. So that then you're getting maybe to a 2, 3, 4 different particles, or 4 different formulations that you can test further. And you can then start looking at, well, am I really getting the outcome that I want? And we can see that if we take these cells, if we take these human mesenchymal stem cells and we put them into 21 days of culture with these particles, that you can generate, you can start to see what? Calcium deposition. So signs that really you're starting to have something that looks like bone, right? There's some minerals being deposited. And then you can look at the different types of biomarkers, things that tells you what these cells are doing. Are they really becoming? Are they going towards towards being more bone cells, or are they becoming more fat cells, or what's going on? So there's certain markers that can help you to test that. And these are, you have early markers, you have late markers, so you can think about what you want to do, again, by understanding what's going on. And you can see that really, without any growth factors or any drugs added, you can really achieve this this type of guided stem cell differentiation into this osteogenic lineage. So that's something very important. That's something that might help with bone healing, or something, understanding how to implant that. Now, obviously, you can think about doing these different types of macrofabrications, doing this in different sizes, shapes, if you want to implant this or this or that. So this just, you can then start expanding your work and start looking at things that will be more interesting to you specifically. And finally, once you do this in some type of a in vivo, you want to make sure that all the stuff that you did in vitro is really going to be translated into some type of a in vivo model. So, right, you don't want to, once you're doing these models, you don't want to be doing 200, 300 rats or mice or something like this if you don't know what is really happening. And I think this is something also important. oftentimes I see researchers, they focus on, doing these steps chronologically. Even though I am presenting them chronologically here, it's not necessary that when you're doing your research that you do these steps chronologically. I think that's something also, that can be misleading. Sometimes, if you really have a hunch, if you really understood what's going on, sometimes you can jump, maybe just with one sample or something like this, and see whether really the outcome that you're trying to achieve, you're getting it. And then you can go back and make sure to characterize this properly and, kind of do the reverse engineering. Oh, did I really have the nanoparticles there? Was the concentration the way that it was? Was it homogeneously distributed, et cetera, et cetera? If you're really getting that outcome. So I think this is something very important, because a lot of researchers, they think they have to do all the steps, 1, 2, 3, 4, 5, 6, 7. Sometimes it's better to go straight for the punchline. Like, what is really the application of this? What is really going to make this work stand out? Where is the innovation coming from? And see if you're really able to achieve that. And then you can reverse and kind of go back and look at all these different steps. And the reason why I mentioned that is because there was another person in the lab. I wasn't part of this work, but they took the same nanomaterial. They took the same gelatin, the same silicate nanoparticles, and they thought, hey, you're having some of this shear thinning property. It's kind of injectable. Maybe we can use this some type of, for embolization, and they were able to publish, this into Science Translational Medicine. And I think this actually is now a commercial product. I think the company was established by, the two lead scientists on this from Harvard, and they established a company. And they got bought out by Boston Scientific, and now this is something which is or one derivative of this is what is actually getting into the clinical application. So you can see really how important it is to understand the different vantage points, right? I was like, oh, this is so great for one thing, but actually, if you talk to your community, if you talk to different scientists in the field, you will find that maybe your nano formulation or something like this has a much better application into something else. So, I was like, well, sometimes I wish I was part of this work, because they published in such a prestigious article, and they moved on into a company and stuff like this. But you can really see that applications can sometimes come from unexpected research and stuff like this. And this is just their work. But when we're talking about bone, it's not just about getting the mechanical properties and doing things which are stem cell differentiation. You also need vasculature. This is one of the biggest problems, one of the biggest challenges in tissue engineering, making sure that you really have good vascularization. And here we thought, well, maybe we can improve that by using some peptides or some other way, some other kind of a small molecule that can signal to the cells. Not only will you become stem cells, but maybe we can include and co-culture a different type of cell that would generate vasculature. So when we actually want to implant this, it can survive. It's not just something that you're holding on into in a petri dish. So here we start to see something that looks like capillary structures and things of that nature. So I just want to make sure that you understand that there's different ways that you can look at this. The same problem can be looked at from different angles. And here we're looking at solving the vascularization problem when you're thinking about bone and stuff like this, not just maybe the mechanical or the stem cells differentiation aspect of it. And we can soak this. We can show that in animals slowly that you can start to get infiltration of new blood vessels, which is very important when you're thinking about implanting stuff, right? So the second part of this, kind of a bone part, I mentioned is the handheld. So this is all great, when you're thinking about it and studying it, understanding it, but 3D printers and stuff like this, imagine, a very bad scenario, so there's a car accident or something like this, the patient has this complex, fracture, and they want to do something with, with these materials, and they want to 3D print them. For something to, for that to be really, functional, you would need to scan the patient, generate an MRI image, take that MRI image, clean it up, generate an STL file, take it, print it, at a facility, sterilize it, all these different steps. This will take maybe days, but tens of hours, at least. So we thought, well, can we solve this problem a little bit differently? can we put something in the hand of a surgeon that could fill that bone defect and help the bone generation right there in situ, right? And we thought, well, that might be possible. We can still take the same nanomaterials that we used before, but maybe we can switch out the polymer, because making it something that's so soft, maybe we can do something which is more mechanically robust. So, what we do here is we take, your extruder. I mean, this is something that is not, I mean, we're not talking about anything high tech, right? It's this regular extruder thing, piston moving around, mixing different polymers with the different nanomaterials that we want. And then maybe we can think about putting that directly in situ and printing inside the patient in situ. So there's essentially 5 different challenges that you can think about. Number one, if you're thinking about 3D printing in situ into the patient, the first thing that's going to ask you is like, what's the temperature, right? So if you look at all these different 3D printers and all the stuff that's going around, you will find out that they print at 200 Celsius, a lot of these polymers. So if you actually try to print into a patient at 200 Celsius, you're burning them. You're not really helping out anything. You're maybe making the thing even worse. So the first challenge is, can we find something which is low melting, a polymer that is low melting? Can we do something which is biodegradable? We don't want to remove it. Once we do this thing, we want to kind of let the healing process, continue on. Can we think about things which are, if we're talking about surgery and stuff like this, maybe it needs to be antibacterial, so decrease some type of infection and stuff like this. It has to be mechanically strong. We don't want to put it there, and like these gels, what happens with the gels if you put half a pound of muscle on it? It collapses. So we need some polymer that is going to be mechanically robust, that is going to be able to withstand the weight that it's going to be put on it. So different types of so all these different types of things. And maybe at the end of it, because we're talking about bone, can it still be osteogenic? Can it still help the bone get those those cells that are around, from after the fracture or whatever, that would help with the bone healing. So we do this. We kind of reprogram the 3D pen. And then we thought, well, can we get it to print? And you can see here, maybe at the very exit of the tip of the 3D pen, you can have things that are maybe 60, 70 degrees, but as soon as it's as it's coming down and comes in contact with the tissue, it cools off very rapidly. So you can think about this even maybe for dental defects, right? Maybe there's a bone defect inside, maybe there's an infection or something like this. And you can think about actually filling that defect right there. So the benefit of this is also that it kind of stays in place. It kind of interlocks mechanically as you're filling all these, holes, all this defect. It kind of gets stuck inside, that bone, and it stays right there in place. And you can think about adding a camera there, maybe even a robotic arm in the future. At MIT, they're already doing this. And maybe a guy, in Jeddah is operating or filling a defect for somebody that is, halfway around the world, and so you can think about all these different things, how they quickly can add up, really. So we think about different, compositions of hydroxyapatite and zinc oxide with the PCl to generate these things. Why? The zinc oxide, we wanted to, because there are some reports that zinc oxide could be could play in that, osteogenesis or osteoconducting, if nothing else. And it's antibacterial, right? So you can think about that, and we can see, okay, it's getting there. We're working there. We can look at guiding this osteogenesis in vitro. If we put cells on top of this, can we get the same effects like we did with the gels? And we can see that is kind of occurring. And now our future work is to actually do this in an animal study, right? To generate a bone defect, maybe in a skull of a rat or something like this, and fill it with our material and see whether we're getting all this, effects that we thought we could get in vitro. Can it really happen in vivo as well? Maybe it could be applied, like I mentioned, in dental applications. Maybe there's different membranes that have been proposed before, but maybe we can think about this being printed directly into the defect. Okay, so that's kind of the first part, and I don't know. I'll try in the next few minutes to really try to go through all these I guess there was a lot of questions. I didn't pay attention to the chat as I was talking. I got carried away. There was a lot of questions. So I'll try to go through this really quickly so we leave enough time then for questions. And if you asked your question before, because I see there was like 100+ messages in the chat, I probably did not see it. So at the end of the talk, I'll give you an opportunity and you can ask them one by one. But really, the other part of the talk is thinking about immunotherapy, right? Immunotherapy is an important cancer treatment approach, as a method, right? So you can get highly specific immune response, minimize the toxicity, make it long-term immunity. So this is something that was, kind of initiated by Glenn Granoff, I think, he was at Harvard, now he's at Novartis, and another lab. I think I will cite his work in Mooney's lab, okay, where they kind of started this kind of the idea of this GVAX. They thought about they could take genetically modify these, immune cells to secrete these, immune stimulating factors to basically tell the immune system, hey, this is a cancer cell. But taking this and doing this genetic modification and implanting it back into the person, these dendritic cells, finally the phase 3 clinical trials were unsuccessful. It was not shown to be effective treatment approach. So can this be overcome by generating let's say a 3-dimensional scaffold? If we can basically train and guide stem cells to become bone cells, why can't we do the same thing with immune cells? Why can't we take and generate these, kind of immune responses by a 3D scaffold? So this is, like I said, Dave Mooney's lab at Harvard that really talked about and initiated this bunch of work in Nature and stuff like this, where he's able to reprogram cells in situ in the material. He's able to kind of basically do a boot camp, right, for the immune cells, where they come into this scaffold, they're able to be trained, and so they understand that this is a cancer cell, and they can go out into the rest of the body and destroy those cells. So what we want to do is train the immune system to recognize cancer cells, deliver it in a minimally invasive approach, so no surgery, so that it could eradicate and even maybe prevent tumors from having some type of a reoccurrence or something like this. You want to have, but what's important? What are the technical challenges, right? When I talked about the bone stuff, I gave you like 5 different factors, has to be low melting and all of that. So again, what problems are you solving? How are you approaching this? So you need something which is microporous, right? So cells have to be able to infiltrate into this. So they have to be elastic, right? So they can be compressed and injected, right? So all these different type of kind of engineering, components have to be incorporated into these ideas. So, and one of the hallmarks of cancer is that they're hypoxic, right? So the cancers are growing so quickly, they essentially start to eat all the food and they consume all the oxygen that is around, so that maybe if we can make that also oxygen releasing, maybe we can change that tumor microenvironment where the cells are able to function, the immune cells, right? So what happens is the immune cells, they come in, in a cancer, they say, there's no air, right? It's hypoxic, they can't function properly, and they leave. And that cancer is able to then go further and further. So maybe we can add oxygen in there, maybe we can modulate that tumor environment and rescue that activity of the immune cells. How we fabricate this. So this technique was basically initiated by Ben Sharif, Sidi Ben Sharif. I think he's at Northeastern University. So we learned a lot of this technology from him. I got to give him also props for, getting us involved in a lot of this stuff. But essentially the main component is that you're doing cryogelation, right? And cryogelation or cryopolymerization essentially means that the aqueous solvent that you use to dissolve your polymer, it freezes once you put it in freezing temperatures, right? So what happens is that then that chemical cross-linking reaction happens only at that interface, where of the ice and that polymer phase, and essentially you get pores, because once you melt that ice, the chemical reaction couldn't take place there, and you're getting these large pores and very thick interconnected polymer walls, which allow for these shape memory properties, which allows for this injectability, very unique mechanical properties, right? So you modify the polymers, you do cryogenic treatment, you thaw it, you get all these different, and you get these very unique, very highly porous structures, right? And that it's syringe injectable. So how do we then we can think about doing this and getting that oxygen release component part of it. And like I mentioned, these T cells that would usually be inhibited, in the oxygen releasing cryogel, they would actually start to function the way that they should. And I think you can see this if there's no oxygen release versus if there is oxygen release, you can see immediately those bubbles. And this is where I talk about, kind of a jumping and, testing your hypothesis, right? So you can see immediately, you don't have to do the FTIR and all this other stuff. You're already seeing those bubbles. So you can see that your work, your idea, your hypothesis is really working, that you can kind of generate something, oxygen or something like that. And, like I said, you can go back, you can characterize, if you're using calcium peroxide, like in our case, you can look at, for these calcium stains, like alizarin red, which will tell you how much calcium you have. You can look at different methods essentially to show you whether you're really having what you claim that you have, which is fine. And you can look at your oxygen release over time. Really, are you able to change the microenvironment of that from being hypoxic into something which is more normal or normoxic, right? And what you want to do, and this is a very, very intensive slide, but I think the video will sum it up. Without oxygen, you have these immune cells. They're walking right by the cancer cells. They don't really care that the cancer is there. But if you have those same immune cells with oxygen, they immediately are able to recognize that as a cancer cell and eat it up. So with oxygen, you're able to rescue that immune function of the cells and get the T cells to kill the cancer cells. Okay, so that's and again, if you look at this in mice, now you can think about implanting it and all this stuff, and you can essentially show that the mice are able to survive much longer when they have this O2 cryogels and stuff like that. You can imagine now doing that. This is now without any, there was no boot camp, there was no training, right? This is just a material characterization. But now if you add cells inside of this, perhaps you can also generate even more and better results, and maybe 100% of the animals could potentially survive and really generating a cancer vaccine. And this would essentially be a personalized cancer vaccine. So once the patient develops the cancer, you can take those cells out, right? You can then irradiate them and put them right back into the patient, right? Because now as irradiated, they can't multiply anymore. They're inside the scaffold, they're at the right place, but the immune cells can come and recognize it. You can do these things to help it, help the patient. And this process is maybe one day, as opposed to the previous GVAX technology, which would have to do all this, explant them and do the genetic modification in a lab. And again, something that takes on weeks, as opposed to something that they can take on days. So you can generate these O2 cryogels with catalase. So catalase is very important because we're using calcium peroxide. So we want to get that, calcium peroxide is going to go to hydrogen peroxide, and you need that catalase to decompose it into oxygen. You can see that you're able to reinstate the T cell mediated activity, even though it was, because of even in hypoxia. And really, you can use this kind of as a platform now for many solid tumors that could really, push forward this idea of cancer immunotherapy. So I will end there, a little bit abrupt. I tried to go a little bit fast through all these slides to make sure that I cover most of it in the time. I didn't want to go over. I wanted to kind of stick to the 45-minute time frame. I definitely want to show all the acknowledgments from the center, Dr. Ahmed Shahriar, Dr. Ammar, who are the director and the deputy director, but also collaborators like Ali Khadam Hosseini previously, who was at Harvard, now at Terasaki. Ali Tamayel, we work very much with him. A lot of the work going on with the bone and the handheld printing is in collaboration with his lab at UConn, University of Connecticut. We worked with Professor Argia Paul, who's in Canada. So that work initially with the gels and the cells, and then other people that we still collaborate very well with, like Dr. Anwar. I used to make a joke. I started writing this, and all the people's names started with an A. I was like, wait a second, there got to be others. So Dr. Noman, who's also in the Nano Center, Dr. Farid Ahmed, also at KU, Siddhi bin Sharif. We learned a lot from him. Really, really great scientist. So thank you for your time. And now, if you want to put some questions on here, feel free to go ahead. I will open up the chats, and if you have questions, we can.
Yes, thank you very much, Dr. Adnan, for this wonderful presentation. We do have a few questions in the chat box. If you have time, we can take them.
Okay. Okay. So I see one that says, if we're going to use nanomaterials commercially, it means we will be doing massive production of nanoparticles. Is it worth the risk to our environment? We still don't know about the long-term effects of nanoparticles. There are cases in which nanotoxicity has been documented. So what's your take on this aspect? This is a great question. This is a very valid concern. Toxicity is very, very important. And we have a very good review that we wrote on, the toxicity of carbon nanotubes, because we did a lot of work with carbon nanotubes. So number one, we have to think about nanomaterials is very broad, right? So and we talk about polymers, it's also very broad. There's natural polymers, there's synthetic polymers. There's the nanosilicates, they're actually minerals. The laponite, it's a mineral that you find, on part of a mountain mining, right? So not everything necessarily means that it's synthetic, and not everything means that it's automatically toxic. But it's very important to do those types of studies. And I think that's why, for example, this whole idea of organs on a chip, where you can do a lot of things, a lot of testing, where you can generate kind of an organ on a chip that would mimic the structure of a human tissue, or some function of that tissue, will help you kind of screen through that. So I think, it's extremely important. Like, for example, if you're going through FDA, they will not even entertain an idea of a material unless you go through their lab. Just like you can do all this published stuff and you can get all these different patents, but if you want them to approve your product, it has to go through their lab that they believe in their results and stuff like this. So yeah, I think I'll leave it at that, because it's a very broad question. It's like risk to the environment and all these nanoparticles. So it's not very specific. If you have a very specific nanoparticle that we want to talk about, we can discuss it. But there's things that are toxic, and there are things that are not toxic. And it also depends on the concentration, right? If you drink a lot of water right now, if you drink like 2, 3, 5, 10 liters of water, you probably will die just from the water. So what do you have to kind of define all these different things in terms of toxicity and stuff like that. Okay, so let's see, next question.
Yes, sir. So we have another one from one of the participants who's asking us, can we use the nanoparticles, especially cerium oxide nanoparticles, as nanofertilizers in different crops to improve the plant growth and nutritional quality? Yeah, that's
yeah. So that's a very important question. So actually, this was something that I came by. I was actually asked to review an article that was all, again, on carbon nanotubes, and they showed how carbon nanotubes can actually increase crop yield, and the carbon nanotubes can improve like the size of the tomato. And then, like, we grow little tomatoes, but we add carbon nanotubes, very small percentage of it, and then the tomatoes become huge. So I was very surprised by all these nanotechnology applications in agriculture. And I think that is something which is very, very, very interesting. Something very interesting, something that I haven't thought about before, but you can really see that even nanotechnology is getting into those types of, fields. So I worked very briefly with cerium oxide with one lab in Kaust. He was an Italian professor, or he was Enrico Traversa. So he worked with these cerium oxide nanoparticles, and he showed how effective they were for cardiac cells, to actually prevent some type of a, ischemic damage or something like this. So I don't know if it would be suitable for agricultural applications, but I know that there's definitely, that's why it's always important to go back at what is what is the, what is the reason why using these nanoparticles? Do they have some antioxidant activity? Maybe that antioxidant activity is the reason for, for the outcomes that you see. So for example, a lot of the work that that I presented was really taken much further than what I showed here by Akhilesh Gahawar lab, I think at University of Texas A&M. So he worked a lot with these nanosilicates, and he showed what is the actual mechanism? Why do these, mesenchymal stem cells really become, why do they go towards the osteogenic lineage? And they show that this could be something by activating reactive oxygen species and different pathways and stuff like this. So it's very important to understand, okay, I see these unique properties, but what's unique about them that is going to be relevant to my application? So maybe the cerium oxide could be something that is, preventing some oxidative damage, or maybe it's promoting some oxidative damage that then helps the cell or the plant grow better. Okay, I see using nanoparticles as MRI contrast agents. Definitely. So that's a lot of work. I know a lab at University of Chicago or University of Illinois Chicago, where they're trying to kind of use dual contrast agents. So something that can be used as a contrast agent in MRI and a contrast agent in CT scan. So this is something which is really interesting. So then you can think about those different applications, and we have we have a very nice review on this maybe a couple of years ago on these magnetic nanoparticles and how they can be used. So essentially that's what you're talking about, right? When you talk about MRI, so you want to generate magnetic nanoparticles that are maybe super paramagnetic or something like that, right? Okay, let me see any other cerium oxide. Okay, nanofertilizers. So yeah, it definitely it could possibly be used, cerium oxide. Let's see. Nanotechnology for gene manipulation. Can we use nanotechnology for gene manipulation to cure diseases and target specific gene defects? Definitely. There's a lot. I mean, there's a whole array of. I mean, the whole idea of the mRNA vaccine, right? So, mRNA vaccine is essentially doing something to that effect, right? It's not you're not necessarily manipulating the gene, but you are kind of getting things to be expressed, right? That normally wouldn't be expressed, right? As a way of generating a vaccine. So I'm sure that technology could be then expanded to maybe some CRISPR-Cas9, to other different ways. So that's definitely that's definitely possible. This is a very broad question. I'm sure you can do a whole seminar on nanotechnology for gene manipulation. So there's basically using the nanotechnology as a way to deliver the genes. So gene therapy and all these different things, right? Can we use nanoparticles for replacing or treating atherosclerotic arteries? Atherosclerosis. Well, so essentially what you're having there's actually a publication that comes to mind where they used nanomaterials that were sensitive to the changes in pressure, right? So when your arteries are starting to get clogged, part of it, part of the artery is then the flow in that artery is different than the rest of the body, or in the areas where there is no clot, right? So then if you have nanoparticles that are sensitive to those changes in pressure or whatever, they can release drugs. So there was a famous paper by Don Inberg, maybe, I don't know how many years, but a few years back, where they did this. They generated nanoparticles. It was published in Science and stuff like this. So it's very, I don't see that would be something very, very difficult. Yeah.
Thank you very much, sir. Due to time constraints, we can take just one last question. Okay, so can nanoparticles be used to monitor specific metabolites in the metabolomic studies? The last question we're taking.
Yeah. So this is if you know Google X, This is exactly what Google X, or now Google Life Sciences, what they try to do, and this is exactly what they're trying to do. They're trying to have nanoparticles that will be constantly kind of circulating through your body and monitoring for changes. Now, those changes could be maybe changes in pH, maybe it could be some, like more physical changes, but it could be, I don't see a reason why that couldn't be something with a metabolite, right? Where maybe there would be metabolite would bind to the nanoparticle, and then the nanoparticle is going to change its color, or it's going to change some other property, and then you have some type of an external detector that's maybe on your wrist that can, sense the flow of those nanoparticles and be like, hey, something's going on here. So definitely, that's not science fiction at all. That's something that is people are working on.
All right. Thank you very much, Professor Dr. Adnan, for this wonderful, very interesting and very informative presentation. We can see lots and lots of praises from all the participants in the chat box. We are very grateful to you for sparing your precious time for us today. Also very grateful to all the participants who joined us today. Everybody who's asking about the recording, yes, all of our webinars are available on our YouTube channel within 1 or 2 working days of the event. So you'll be able to access the recording from there. Thank you so much, everyone, for joining us. And we wish to see you in future important events. Thank you very much, sir. Thank you and goodbye.
Thank you. Bye-bye.